Hi, my name is C Uruena, and this is my presentation on biochemical hallmarks on cells undergoing apoptosis. So Y chromatin packing is created upon DNA fragmentation. You kind of have to know a little bit of background to understand the relationship. So nucleosomes are packed together to form chromatin fibers and chromosomes. The chromatin actually makes up a chromosome consisting of DNA and protein, and the histones are the major proteins in chromatin. DNA is packaged by forming coils of nucleosomes, and the DNA fragmentation breaks these DNA strands into little pieces, which can be seen in figure 1 of the DNA run on agarose gel. A classic feature of apoptotic cell death is chromatin condensation DNA fragmentation. In dying cells, the DNA is actually cleaved by an endonuclease that fragments the chromatin into nucleosomal units. This cell death is associated with DNA fragmentation. Just again, from figure 1, you can see that. The enzyme responsible for apoptotic DNA fragmentation is the CAD, which is inhibited by the inhibitor CAD, so ICAD. The enzymes most widely linked to the apoptotic cell death are caspase enzymes. So this DNA fragmentation takes place because of this. And also the apoptotic population appeared to be derived possibly from G1 and S phase cells, not from G2 or mitosis. This coincides with the cell cycle compartments in which DNA and RNA polymerases function. So some of the roles of proteins. Nucleosome is the basic unit of chromatin structure packed together to form the chromatin fibers and chromosomes. The linker DNA is the DNA that is degraded during digestion from 200 to 146 base pairs in length, and the DNA associated with the core particles measures about 146 base pairs, which is enough to wrap 1.7 times around the particle. So the histone H1 is thought to bind to the linker DNA and facilitate the packing of nucleosomes. Cytochrome C is a heme protein that is localized in the mitochondrial interspaces, and they're essential in mitochondrial electron transport, and they also intrinsic type 2 apoptosis. The mitochondria is actually what triggers apoptosis when it releases cytochrome C into the cytosol. Apoptosis is actually a mechanism in which the cell recognizes that it's damaged and it basically destroys itself. So it's a process of the programmed cell death. It involves a specific series of events that leads to the dismantling of the inner contents of the cell in which the DNA fragments. This forms the diagnostic ladder of fragments. And then one of the proteins that's related to it is proteins like PUMA, which promotes apoptosis. Caspase is enzymes that are cysteine proteases and key mediators of apoptosis. They play an important role in apoptosis as they initiate the signal and carry proteolysis, leading to apoptosis. Endonuclease are also enzymes. So they're enzymes that degrade the nucleic acid like DNA, cuts the molecule internally. In apoptosis phases, the cell's DNA is cleaved by apoptosis-specific DNA, endonuclease, or DNase, at regular intervals along the DNA, resulting in DNA fragments. So nuclear condensation, this is one of the biochemical hallmarks of apoptosis that I kind of did a little research on. And in addition to caspase activation, cytochrome C release, and fragmentation, this is another biochemical hallmark of apoptosis, the nuclear condensation. This research article that supports this was conducted to understand how chromatin condensation paralleled by DNA fragmentation is one of the most important criteria used to identify apoptotic cells. This experiment involved the use of confluent cultures of an embryonic fibroblast cell line that was induced to undergo either apoptosis at low serum conditions or mitosis. In the apoptotic cells, the chromatin condensation is accompanied by degradation of the lamin A plus B, as it was observed that the filamentous structure of the lamin network disappears during apoptosis, and it was also time-dependent degradation. The chromatin condensation during apoptosis appears to be due to rapid proteolysis of the nuclear matrix protein. So as seen as figure 3, this was a figure from the experiment. You can see the electron microscopy of a newly formed apoptotic cell that they actually induced with the low serum, sorry. And then the other experiment or research that I went over was the phagocytosis of another cell, which is also, aside from the previous mentioned hallmarks of apoptosis, it's the phagocytosis by another cell, which the research article that supports this was conducted to see the relationship and mechanisms by which macrophages interact and respond with apoptotic cells. The macrophages plays an important role in the clearance of dying and dead cells and sites of histologically visible high-rate apoptosis. The association between the two shows that very often the apoptotic cells and its fragments are visible in phagocytic vacuoles within the macrophage cytoplasm. For this reason, the macrophage is considered widely to be the professional phagocyte of the apoptotic cell. A famous example is the apoptosis occurring during the development, like the cells of interdigital webs deleted during the limb remodeling, which this is actually what is showing in the figure 4 to the right. It lets you see the engulfment of apoptotic cells in situ. In this research, macrophages display an interest in sites of high-rate apoptosis where dying cells reach the stage of histological visibility. Therefore, it plays an important role in clear up operations at sites of apoptosis, and it's critically important for regulation of the immune responses. Much of the current knowledge behind this hallmark of apoptosis actually comes from molecular cell biological studies of macrophages interacting with apoptotic cells in vitro. And that's the end of my presentation. Thank you for listening.